Also on Thursday, we see a more friendly face that we haven't seen before. It's Dr. Ballard, and she'll be joining us to. I don't exactly know why she's here, but she asked to be here on Thursday, so she'll be joining us, and I'm happy to have her. So again, I can't find anything in my notebook, so okay, we'll just figure it out later. That means that if I say something wrong, you tell me, right? Are there any questions so far? No. Okay. So, translation and protein synthesis and viral genome replication and then release. Okay. So let's draw the mRNA, but let's draw the viral genome in more detail. Okay. This is basically what it looks like. So here's the plus strand RNA, and by the way, there are ribonucleoproteins that bind, right? That continue to bind. Okay, so you plus other ribonucleoproteins that bind. So the attachment entry and uncoding occurs, and the genome is released from the virion into the cytosol. One thing to remember is that there are RNases that would be very happy to degrade any mRNA. So therefore, the two ends are protected, and we'll talk about the protection. Okay. So therefore, you don't want anything happen to this. Number one. Number two is pay attention. This is poly A. The poly A tail is actually a part of the coding region. The poly A tail is not added on by polyadenylation. It is a part of the gene. Okay? So therefore, when the minus strand and plus strand RNAs are being synthesized, that the poly A region here will convert into poly U's, right? So this is 3', that means it's 5'. Right? So that's very important to understand. Okay? And there is a very specific, it's roughly something like, I don't know, 50, 60 or so nucleotide long. Okay? So far so good. Now, once in the nucleus, in the cytosol, in order to start translation, a couple of classification of host cell proteins will bind. So you will have binding here and binding to the. So this structure right here is called the clover leaf structure. It has a number of functions, a couple of functions, and then this is called the pseudoknot. It also has a function. I'm not just telling you these words because they're fancy and I want to show off. Okay, they have a function. So to the clover leaf, you will have proteins that are called poly-RC. C stands for clover leaf, binding proteins. And a few of them. So these are tiny, tiny proteins that bind. To the poly A tail and somewhat the pseudoknot, you have the poly A binding protein. So you have that going on. All right, and then translation occurs. Translation occurs because and if you remember anything about translation for a cellular for host cell eukaryotic cell, what do you have to have? What must the structure of the mRNA be? Circular, right? So this have to interact with this. So that's what the poly-RC binding proteins and the poly-A binding proteins do, is that they bind to the respective end, and then they actually have affinity for one another to match the translation, because the small ribosomal subunit that carries the tRNA can only recognize the RNA, both the 5' and 3' region together, right? You remember that? So the 5' and 3' region have to be like this. Before, and with proteins binding before the small ribosomal subunit, with the initiator tRNA bind, right? That's the initiation of translation. Okay? So that's the function of right now, the function of poly-RC binding protein poly-A, that they allows the RNA to circularize like this so that the small ribosomal subunit bind. But before the small ribosomal subunit can even bind, that VPG has to be removed, and remember it's due to the ribosome again. I can't tell whether, I don't know, I can't find any information as to whether it's the same small ribosomal subunit or if it's something else. So we'll just say the ribosome, this ubiquitous ribosome that removes a VPG. Yes, ma'am?
I think you said this last class, but that ribosome that removes the VPG isn't necessarily the same one.
Isn't necessary, but I don't know. Right? I don't know.
Okay.
I don't know if it's the same or a different one. So we'll just pretend that, well, I just don't know, so we'll just say hand waving that somehow the ribosome is responsible to remove that. But beside that, translation occurs in this way. How's that? Okay? All right. So translation here is cap independent. Translation initiation. And for cap independent initiation, what is required? An IRES. Right? That is related to a secondary structure. Just happened that the clover leaf is a secondary structure. Okay.
So is it right next to the clover leaf, or is it?
Yeah, right next to. Some of them are right next to, others are found within, but that's not as important as the fact that the IRES is right there. Okay. Okay. So that's important because it's cap independent. Most of the translational activities in your cell is cap dependent. However, the mechanism, cap dependent and cap independent translation initiation use roughly similar, not the exact same thing, but similar mechanism. So if you remember, how about if I go through translation and then come back to the regulation? How's that? Because it actually is relevant to the virus. So cap independent translation is basically having a small ribosomal subunit which is already bound by an initiated tRNA carrying that methionine, right? Binding to the IRES, which in turn is already IRES, which basically the 5' and 3' ends are already interacting, right? And so the RNA, the small ribosomal subunit binds to that whole area. So it's not enough to just have the RAS, but the 5' and 3' ends have to interact before a small ribosomal subunit can be recruited. Small ribosomal subunit then looks for the AUG, the start codon, right? Which should be right next to it. So the start codon should be right there. So there's no scanning. So AUG start codon, and then synthesis of the poly. Even though it's called the polyprotein, but it really needs to be the humongous polypeptide first, and then folds into the polyprotein. Okay, so transcription occurs, and now you have, and then folding and all of that. Now you have. Not transcription, I'm sorry, translation occurs. And then now you have a polyprotein. So that's translation. Simple enough. Translation must occur immediately. Once translation occurs, right, at the very beginning, the cell may continue to undergo translation, but it will get reduced. So things to consider. Number one, translation of viral protein, polyproteins must be reduced as the machinery transition into undergoing viral genome replication. Number one. At the same time, because viruses tend to take over the host system, then the translation of host mRNA will be reduced down to next to nothing as well. And the virus has to be able to induce that. So the virus will then slow down the translation of the host machinery to produce host proteins. So we're talking about two different things, right? Suppressing production of host proteins, but also at the same time, after the initial production of the polyprotein, that even the synthesis of the viral protein needs to slow down. Okay, so those are two different events. Slow down, why? For the suppressing the host translation is one reason, and then the slowing down of the viral protein synthesis there's another reason, right? So they occur in two different times and for different reasons. So let's start with the polyprotein first. Polyprotein, as I explained, is this humongous protein that then gets divided into smaller viral proteins. We won't go through, and it's not completely known, but a lot of the processing of the polyprotein is known. We won't talk about any of that. I'm just going to talk about very specific proteins that are important, okay? The viral proteins that are important. Number one, let me make a list. How about over here? Make a list over here. Number one, VPG is important. VPG is going to be produced. Another protein that is important is 3D Pro, and then 3D Paul, and then a viral protease 2A. Okay, these are the important proteins that we'll talk about. Each one has a very specific function. There are more. We won't talk about them more. We'll just talk about these. Yes, sir. Back there and then up here.
So the viruses create a protease that cleaves the polyprotein that synthesizes the first.
Right. So once all the cleavage and processing is done, you get these proteins, and we won't right from the polyprotein. So right, it catalyzes its own cleavage and all of that. And again, the mechanism is a little funky, but what happens with the polyprotein is that as it gets smaller, each fragment has a function, and then which is different from the end result, which is a smaller. So again, it's a little funky. We won't go through all of that. Just basically go from polyprotein to these. That's all that we need to know. Okay. So then, once these polyproteins are produced, right? What are the function of some of these? Let's go with the 3D Pro first. Pro stands for protease. Actually, this is also 2A is also a protease. This is also a protease. They have different functions. This protease right here is responsible mainly for the digestion and modification of the viral polyprotein. Okay. In fact, 3D Pro is responsible for the synthesis of VPG. Okay. So again, 3D Pro is a protease that actually targets the viral proteins and make sure that they're properly cleaved. Whereas the viral protease 2A actually targets host proteins and I'll go through the mechanism in a minute, but to suppress translation of host proteins. So again, the viral protease 2A actually targets host mechanism to suppress the synthesis of host proteins. Okay? So, yes ma'am.
What is the relationship between the 3D protease and VPG?
3D protease, one of its function is to produce VPG from the polyprotein.
Okay.
In fact, 3D Pro basically is responsible for the production of most of the viral proteins, but very specifically VPG. Okay. So on that, VPG we'll talk about. This is the actual RNA dependent RNA polymerase, which is abbreviated capital R, small d, capital R, capital P. And this is a viral version. Your own cell, you actually have RDRP as well. Okay, so this is the actual enzyme that ultimately will produce, will replicate the viral genome. Okay, so far so good. So here is the sequence of events after translation occurs. The initial translation occurs is these proteins are going to be produced. All these proteins are going to be produced. One of the various things that happens is that, again, the viral protein, viral protease 2A suppresses synthesis of host synthesis. How does that happen? Okay. In order to talk about that, we have to talk about the fact that most translational event of host protein occurs as a cap-dependent translation. That means that the cap is required. Okay? And when the cap is required, here's what happens is a cap, 5' poly A, 3'. The cap is going to be recognized and bound by a dimer. It's a EIF 4E and 4G combination. Those of you, hopefully you remember, okay? G, okay? This dimer then binds to the cap, okay? And then the 5' and 3' also circularize, so the mRNA also circularize. And then the small ribosomal subunit not only recognizes the fact that the 5' and 3' are interacting, but also there's the EIF4E and EIF4G are also there. Right? And again, there's more to the story, but that's enough. Okay? And so what the viral protease 2A, what it does is that it digests, it actually prevents this dimer from forming. It mainly actually digests the EIF4E version and leaves the it actually digests the EIF4E and leaves the EIF4G alone. And so when we say that the viral protease 2A suppresses synthesis of host protein, it's not all host protein, only host proteins that have to be synthesized through cap-dependent translation. The synthesis of some host protein can occur through cap-independent synthesis as well, translation as well, and those proteins can still be produced, right? Again, we're only talking about the fact that the protease 2A inhibits cap-dependent translation. Yes, sir.
Okay, just trying to remember back here, but so with 2A inhibiting cap synthesis, that's going to put the cell in a state.
No, it doesn't inhibit cap synthesis. It inhibits cap-dependent translation.
Yeah, it inhibits cap-dependent translation. It's gonna force the cell into like a state of emergency, where the cell's going to realize it's not producing enough proteins, which will force it to go into cap-independent.
The logic is reversed. The cell, by going into cap-independent translation, doesn't mean that the cell is in an emergency. Doesn't mean that it causes the cell to be in an emergency. It's that the cell is in an emergency condition. That's why it turns to cap-independent translation. So what you said is the reverse. So by removing this has nothing to do with emergency, right? The cell. So does that make sense? So
yeah.
What you had is just reversed, but that's okay. Not a big deal. Yes, sir.
So can you describe the difference between cap-dependent and independent role classes again?
Cap-dependent requires a cap. Cap-independent requires an IRES and not a cap. For cap-dependent translation, both EIF4E and EIF4G are required. For cap-independent translation, only the punchline is only EIF4G is required. So what the viral protease 2A does is that it knocks out EIF4E, right? Therefore, but not EIF4G, and in fact, just so much more information. In fact, the cell actually, at this point, increases the expression of EIF4G, but that's a completely different pathway, okay? But regardless, without EIF4E, then EIF4G can interact with this 5' region right here of the mRNA that contains IRES, but in fact it interacts with the actual clover leaf structure, right? And then the cap cannot be recognized or bound at all, so therefore you will not have the small ribosomal subunit being recruited to the cap, but they actually will be recruited to any mRNA that contains an IRES. Yes, ma'am.
Is that EIF or EIE?
EIF4G.
And
This is EIF4E.
And the one under
And
that?
EIF4G as well.
You write EIE.
Oh, thank you. There you go. Picky. Thank you for testing. Thank you. All right, are we good with that? So that's what the viral protease 2A does, and that's what happens. So now your cell, the virus has taken over the host. That's what is meant by the virus takes over the host machinery. It really doesn't take over. It just knocks out and prevent the host from doing its own thing. So now it's geared toward taking care of the viral. So does that make sense? So it's really, that's a misnomer. That's the virus really usually doesn't take over, right? It just redirects. All right, so now you have that. So that's more synthesis of the polyprotein occurs, more of these, okay? And then when you have enough proteins and enough of the 3D protease, what they do is not only 3D not only would modify the polyprotein, but will start to digest the poly-RC as well as some of the ribonucleoprotein. And the important thing about the ribonucleoprotein is that, remember, not really, okay. So the ribonucleoprotein is important, and we'll get to that. Okay. So far so good. By digesting the poly-RC, what happened is that the 3' and 5' ends don't necessarily interact as well, right? So now you reduce the chance of the viral genome being used for translation. So you're reducing the chance of carrying out more polyproteins. Now the cell is, or the machinery is preparing for viral genome replication. Okay. Why reducing and not removing it completely is because, number one, that original plus strand RNA may need to be used again for viral polyprotein production, and any additional plus strand RNA may also be used for polyprotein production as well. Okay? Not just for packaging and producing the virion, but also the process of polyprotein production continues at a lower level. At a lower level, but not at a high level, because you don't have enough of the poly-RC proteins that bind. Okay? Yes, sir, and then over here. Yes, sir.
Yes, so how does the virus reduce the translation again?
Reduce the what? I'm sorry?
Translation. How does it do? What's the, I didn't understand.
3D Pro digest the poly-RC protein.
Okay.
By digesting the poly-RC protein, then that slows down translation. Yes, sir.
So how do you poly-RC be added on to the new viruses during packaging, right? So you
They're
still
attracted to the clover leaf. So all of the, later on when we talk about plus strand RNA molecules being produced, they will automatically form clover leaf, and they will automatically form a pseudo knot. They will automatically have the poly A tail already there. So these three structures right here will be found in all plus strand RNA. Yes, ma'am.
So, does the replication happen at the same time as the reducing of like the host genome and?
Okay. No.
Okay.
Yes, something like that.
Or does it
yeah.
have to wait for these to slow down and then replication start?
By reducing the amount of poly-RC that binds to the clover leaf, that is slowing down. That is slowing the process down. So one affects the other. Digestion of the poly-RC proteins, right, slows down the translation. Because remember, that molecule can be used, can be bound by small ribosomal subunit to initiate translation over and over and over again, right, until the poly-RC producing a lot of polyproteins, until there's a threshold amount of 3D Pro that are present, then they start to digest the poly-RC, slowing down the recruitment of the RNA small ribosomal subunit. And again, so to imagine how this happens, right? So I didn't mean to go into this much detail, but imagine the binding, no binding is permanent unless you have covalent bond. So the 3' and 5' end does this, and then it comes in and out, comes in and out. Right? The more poly-RC protein that are produced, that bind, the longer it stays here, and then it opens up and comes back again. When you reduce the amount of poly-RC protein, binding protein, you're actually allowing for the release to stay there longer before they bind, but then won't bind as long as the release. So then the chance of the small ribosomal subunit catching the two ends being together is less likely. That's how. So again, that's how that works. Okay. Yes, sir.
So you said that the structure of this positive sense RNA is the same structure with all positive sense RNA viruses.
Yes. But not
so-
minus strand. Well, somewhat the minus strand, but not completely. How's that?
So HIV has composed of a genome of 2 positive
Yeah.
sense RNA strands, so it's the same sort of mechanism.
And no, HIV is actually different because HIV uses a reverse transcriptase instead of an RNA-dependent RNA polymerase. And so the plus strand, actually for RSV, there are 2 molecules of the plus strand. That in itself is a strategy. It doesn't use this at all, by the way. So completely, when we talk about HIV, cross this completely out. Okay. So far, so good? All right. I didn't mean to go into as much detail. I thought I can just gloss over this. So cap dependent versus cap independent. Now you have cap independence. Okay. Now the machinery is going to transition from translation into transcription. Okay. So now let's look at transcription, or at least RNA synthesis. So I'm going to keep this. If you want to redraw this, that's fine. I'm going to keep it. Yeah, well, let's see. What to keep, what not to keep. Okay. So, there are certain characteristics that, and how should I explain this? DNA for host cell, for eukaryotic cells, synthesis of DNA requires very specific components, versus synthesis of RNA synthesis of host cell requires other components, right? DNA is our genome. RNA is not. But when we talk about the viral genome, which the RNA molecule itself is a genome, even though it's not DNA, it's RNA. So some of the mechanism that we use to replicate, to synthesize our DNA, right, which is different from RNA synthesis, some of those characteristics actually are adopted by viral genome synthesis. Does that make sense? Even though we're talking about